How does your brain learn about and navigate the world? What brain areas are responsible for your ability to form mental maps of places and spaces? How are attention and memory implemented in the brain, and how do they contribute to learning? In this episode of the Thinking Tools podcast, I speak with Artem Kirsanov, a YouTuber and neuroscience undergraduate student who's produced a number of visually striking and deeply informative videos that answer the questions just posed. Now, in addition to that basic neuroscience, Kirsanov has even more videos about strategies that allow you to learn just about anything more efficiently. Artem is currently a third-year undergraduate student at Lomonosov Moscow State University studying biophysics. His thesis research combines experimental and computational neuroscience, and it's dedicated to uncovering the mechanism of generation of theta rhythms in the hippocampus. Now, he's also involved in a project on building detailed computational models of interactions between neurons and astrocytes as an attempt to understand the role of astrocytes in neuronal dynamics and information processing in the brain. Make sure to check out Artem's channel. It's Artem Kirsanov. I'll have it linked in the captions below. And without further ado, I bring you my conversation with Artem Kirsanov. All right, so can you tell us a little bit about yourself and maybe why you decided to study neuroscience and computational neuroscience in particular?
Sure. Hello, everyone. My name is Artem, and I'm currently a biophysics student in Moscow State University. I'm a third-year undergraduate, so I have like 1 year left in this program. And even though the formal name of the field of study is biophysics, I'm mostly doing my research in computational neuroscience. And now, if I think about it, I have been interested in neuroscience ever since I was really, really young. And for some reason, that picture has always, I always found it mind-boggling that all of the stimuli, like all of the visual information, everything we hear, taste, smell, all the touch is represented in some way in like this array of electric signals in the brain. And ever since I was a kid, I thought it would be really great to decode that, and maybe we could extract information from the brain just by looking at the activity. And what's even cooler, we could inject like new activity inside the brain. So it would be a total virtual reality, like it's supposed to be. And that was kind of my big dream. But obviously, the further you go into the research, the more specialized you become. But I still bear this picture in mind, and I hope to someday dedicate something towards solving this real mystery.
Wow, yeah, that focus really shows up in your videos in the way that you explain things on YouTube. So I'm wondering what led you to start a YouTube channel, and how did that come about?
Yeah, sure. I started my YouTube channel about half a year ago. Yeah, I think now it's even more than that. I published my first video early June last year. And I think this was more of a creative outlet, so to speak, because at that time I was really immersed with lab research and with all the academic work you have to do as an undergraduate, homeworks, reports, different assignments, classes, that kind of stuff. And those activities were they were occupying 100% of my time. And I felt like I slowly started to approach pre-burnout state that, all of my attention is dedicated to that tiny thing. And I need to find a way to channel the energy to have like a hobby or something, as an outlet. And I always had a passion for video, for filmmaking. As a kid, I actually took some classes on animation and 3D modeling. So like I was already familiar with all this sort of software. And, so I decided to give it a go. And I really enjoyed it ever since. It has been, it really has become what it was intended to be. So yeah, that's pretty much.
Yeah, it's, I mean, your animation skills are really, really amazing. I mean, I just have to say the visuals in your videos are really beautiful sometimes, especially when you're showing mathematical models and things that are hard for, especially for me, someone who doesn't have a very deep background in math, to visualize and understand these things. You do a really good job of both clear language, but also just very descriptive, vivid visuals that kind of stick with you. And so I definitely encourage everybody to check out your channel, watch especially the video on neural manifolds. That's something
that
I feel like we couldn't probably talk. Well, maybe we'll get to that in this video, but that blew my mind, and I had never encountered that before. So anyway, I encourage people to check out that video for sure, and your others. And some of the other stuff you do on your channel is talking about learning strategies and kind of helping people to develop more effective techniques for studying and just learning in general. So it's interesting. I found that an interesting combination where you have both the like really technical, hard neuroscience in there, and then just you kind of expand out to be like, well, how do we apply this? How do we actually use, well, maybe not those specific things, but how do we use what we know about the brain to make ourselves more effective learners? And I really love that. But today I want to talk mostly about the neuroscience side of things. And I thought it would be kind of fun for us to go through maybe some of these concepts that you've covered in some of these videos and talk about them, because they're things that I have not talked very much about on my channel. And so I thought maybe we could start with the idea of grid cells and place cells. And maybe we could start with grid cells and what they are. So I kind of let you take that from there, and we can take it however you'd like.
Yeah, sure. Well, first of all, thank you for such kind words. I really appreciate it. And yeah, we'll certainly dive into grid cells, but I just wanted to clear one thing up. And I think it's actually pretty interesting that it was actually the other way around. So I started making videos on like learning techniques and like how to study. And then I kind of, well, it's a wonderful story behind the neural manifolds video, because I actually did it as a contest entry. So it was a turning point. From this video, I expanded to the neuroscience stuff, but we could, yeah, certainly dive into that a bit later. So yeah, now back to grid cells. Grid cells and place cells that you have mentioned are two components of the spatial navigation system in the brain. So, and there is another component, head direction cells, which I briefly mentioned in the manifolds video, but in a bit of a different context. But yeah, as far as we know, those three components, head direction, grid cells, and place cells, they make up the majority of spatial navigation circuitry. So if we think of neuroanatomy, like where inside the brain this mapping happens, the first region, like anatomically, in case of information flow, we could say that the first one is entorhinal cortex, and this is exactly where grid cells are located. And grid cell is nothing else but a excitatory pyramidal neuron of an entorhinal cortex. And what's so interesting about grid cells is that they are firing, or basically, since neurons transmit information in form of action potentials, right? It's kind of a bit of communication between two neurons. And when I say like firing, it means just that neuron generates those action potentials, or in other words, is the spike. Grid cells, their firing is very spatially tuned. So their behavior is determined by spatial location, where animal is physically located in the environment. For example, if you take a lab mouse, and most of the experiments are done in either mice or rats, it will most likely be a cage. And if you record, let's say from one grid cell, right, you listen to its electrical activity as the animal runs around the cage, you will see that this neuron fires. It fires like crazy, generates a lot of action potentials when the animal is at particular spots of this environment, like at particular patches of ground. And you will see that these preferred locations, where the locations that this neuron really seems to like, for some reason, they would be arranged in a regular hexagonal grid. So this grid is kind of the floor of the cage would be like tiled with this hexagonal grid. And it's a really common pattern in science and in biology in particular. So
the hexagonal pattern, right?
Yeah. And so these grid cells are thought to be some kind of a coordinate system. So you probably know that coordinate system is how we can define position. The most famous one is the Cartesian coordinates, or x and y, in 2 dimensions. And there is a polar coordinate system where your location is defined by the angle and the radius, basically. The distance. And in 3D space, there are other coordinate systems as well, like cylindrical coordinate system and spherical coordinates. And those grid cells, they are thought to represent this internal coordinate system, and it's actually drastically different from our conventional artificial coordinate systems that we construct when we're doing mathematics, for example, because the brain, for some reason, uses toroidal coordinates. Now, it may not be apparent the first second you look at the grid, because it just seems that the coordinate system is periodical. But in my latest video, I actually talk about it in a lot more detail, and it took me several days to get into the paper and really understand what's going on. So I'm probably not making a lot of sense at this point, but the collective behavior of grid cells, if you look at their dynamics, it would be localized on a torus.
Yeah. So just to back up a second,
Sure.
I just want to make sure people don't misunderstand this. So when we're talking about the coordinate system that these grid cells map, and when we're talking about the hexagonal structure kind of that coordinate system, it's not that the neurons themselves are arranged in a hexagonal pattern in the brain. It's that when you look at where the mouse is running in the cage, the neurons are firing such that it's as if the space of the cage has been divided into this hexagonal pattern. Is that the right idea? Okay.
Yeah, and this is actually crucial to understand that, well, for the case of grid cells, there is some relationship between where cells are physically like as tiny little bags of fluid inside the brain are located near near each other, and like they are representative firing fields. These are these patches of ground where the cell is most active. There is some relationship, we can talk about it, but they're certainly not arranged in a hexagonal grid in the brain or anything like that.
Right.
Yeah,
right. Sorry, go ahead.
Yeah. So I was just going to say that if we kind of, well, I'm getting a bit ahead of myself, but when we talk about place cells, and we'll do in a while, yeah, in a few minutes.
Go for it. Yeah, if you want to jump into that, go for it.
Yeah, I just wanted to mention that place cells, they are also spatially, like their firing is spatially tuned, and there we cannot find any relationship like between the locations of cells in the brain and their preferred locations in the environment. So they, at least I'm not aware of it. As far as I know, like all the papers just mentioned that like there is no clear kind of mapping between where cells are located and where they are firing fields are located. But yeah, this is a story about place cells. Yeah, so just to transition to that, grid cells, which, as we said, provide a coordinate system, they project. It means that they send their axons like little wires. So they send information to the brain region called the hippocampus. And inside the hippocampus, there are, like hippocampus also has its own pyramidal neurons. And these are called place cells. And in fact, place cells have been discovered way before grid cells, because as far as I know, place cells have been discovered in 1971 or something like that by John O'Keefe and Jonathan Ostrowski. And grid cells have been discovered in early 2000s by Edward Moser and May Britt Moser. Yeah, and the Mosers and John O'Keefe later shared the Nobel Prize for these, for the spatial navigation system inside the brain. I think it was awarded in 2014. Yeah, so place cells have been discovered first, and here the picture is quite similar. So we listen to the activity of one place cell, one neuron. The animal is running around, and we can see that it's the firing of this neuron is also spatially tuned. So it's modulated by the physical position inside the cage. And this neuron prefers a single patch. So like there is only one firing field of the cell. So place cells, they provide information about the exact location of the animal within the environment. So if you take, for example, one cell, it could be very active when the animal, let's say, is in the middle of the cage, or if it's in the like top right corner or something. Whereas the activity of an individual grid cell, since it's a hexagonal grid, it could, like, it's ubiquitous. I mean, not ubiquitous. The word is ambiguous, I believe. Yeah. Still the second language. Yeah. So yeah, the ambiguous information, because active firing of the neuron of the grid cell can mean that the animal is at any of these, patches that are arranged in the hexagonal grid. And yeah, this is actually how, at least we believe that this is the process. Basically, the firing of different grid cells, they kind of converge, and we can calculate exact location. So we can translate the activity of grid cells into place cell activity, and give and basically read out the exact location of the animal within the environment. And now that was a lot to take in, so maybe we could sort of back up and elaborate on some of the things, because I understand that it was probably way too fast.
Yeah, so I think we should dive a little bit more into what the difference between grid cells and place cells are, because when I first encountered these years ago, I at first thought they were just two different names for the same thing, but they're part of two different systems. Well, one system, but two kind of subsystems of this spatial navigation system in the brain. And they're located in different brain structures, as you mentioned, grid cells in the entorhinal cortex, place cells in the hippocampus. And entorhinal grid cells, correct me if I'm wrong, but they allow us to navigate through spaces. And these could be fairly undifferentiated spaces, meaning like we might not be familiar with the surroundings that much. Yet this is kind of what allows us to form that cognitive map. Or am I now? I think I'm messing the two up. So let's get into the differences between these two. Yeah, take that. Go for it.
Yeah. So, like you said, those are two different types of cells that are located into different brain regions, but I believe they form like those two types. They form one system for like spatial navigation. So even though if we trace the information, it gets first to grid cells, then they project to place cells. But the hippocampus is structured in such a way that essentially information from the hippocampus, like it flows back. So it's a connected system. Like they form a loop. Like it's a
okay.
Yeah. So they communicate back and forth.
And
yeah, and you mentioned the such a thing as a cognitive map. Now, this is a term I believe that was coined eventually. It was coined by a psychologist, right? So it's not like a hardcore computational neuroscience word. It's kind of more when we observe the rat, for example, or a mouse, and we see that it can find its way through the maze, and it can extrapolate what path to take and like how to move around. And we kind of hypothesize that inside its brain there should be like an internal map of this location, all right, of this maze that allows the mouse to navigate effectively.
Right. Okay. So these two work together to allow for our spatial navigation system. Okay. So that I think that gets the idea across of kind of the differences between these two, what they're for. So let's move on to talk about a different aspect of the hippocampus. We've been talking about the place cells that are in the hippocampus and the close nearby entorhinal cortex and the grid cells. But another topic that you've covered on your channel is memory consolidation and hippocampal replay. So this is kind of zooming out from maybe looking at individual cells and talking about how the cortex and the hippocampus interact in the process of memory consolidation.
Yeah, sure. So hippocampal replay refers to the effect that when, for example, I think it's important to understand that each memory, for example, imagine like you are a mouse and you are expected to like find your way out of a maze, right? And after some attempts, you finally find a way out, and like you get a tasty reward, let's say. And the task for your brain is to kind of consolidate that route, meaning when you are placed to that environment, like, next time you should be able to at least remember, or either remember the exact path, or at least find it with much less attempts. Yeah, and this is in terms of this spatial navigation system. But one thing I mentioned about place cells, and this is the thing we are not really clear in our current understanding, is that these cells, they seem to be used not only in like navigation inside a physical space, like a maze or a cage, but rather they exhibit this pattern, that they have this preferred firing field in more abstract spaces. And I described this very brilliant experiment in the video about place cells, that mouse basically was trained to like manipulate a lever, to like push it to adjust the frequency of the sound that was played to the mouse, like in its cage. And the task, I believe, was to, the experimenter first presents, they first present a sound of a particular frequency to a mouse, like very briefly, and then it was trained to get a reward, basically move this lever to adjust the frequency of the sound that's playing to match those two frequencies. Yeah, if you think about it, it's a very very tricky thing to do, I believe, even for a human to match the frequency of a sound. Yeah, and when the mouse was basically pulling, manipulating the lever, they recorded electrical activity from the neurons in the hippocampus. It's the same cells that usually like serve as what we call place cells, that they have a firing that's tuned to physical location. But now the mind-blowing thing is that there seem to be place cells, but in this space of sound frequencies. So if you think about it, as the mouse is like pushing the lever, it's moving on a linear track on in a frequency dimension, right? Like it's a one-dimensional space of frequencies. And so there seem to be place cells. The hippocampal neurons, they prefer a particular range of frequencies. So like that cell like is firing like crazy around a particular frequency, and then another range for another cell, and stuff like this. So this pattern was observed in the more abstract space, which is like, I don't know, the first time I heard it was I don't know, it totally like gave me a completely new perspective on things. And there have been a number of experiments in this fashion, and we now believe that hippocampus is like this machine for constructing behavioral variables and keeping track of them. Because if you think about it, when you are a mouse who's navigating around a maze, in this behavior, the most basically the most important variable to you at this point, or a number of variables, are which encode your position, because you need to find a way out. If, however, you are a mouse that's manipulating the lever, the most important variable to you in this case is the sound frequency, because then you'll either get a reward or not. And so hippocampus is the circuitry that's kind of constructing these variables, whatever is most important to the animal at the current moment, and is used to kind of navigate in this space, to manipulate the variables. And the circuitry seems to be seems to be the same, because it's the same neurons, same cells, and the same kind of place cell-like pattern.
Yeah, that's incredible. I remember, I almost forgot about that. That's amazing. Do you know of any other examples outside of the sound frequency one that where the place cells are recruited for kind of creating or mapping the variables, I guess?
Yeah, I believe it was. I don't know the name of the authors, unfortunately, but it's the concept of it's the concept of a concept cell, because basically, I think it was called. I may be lying here, but probably it was on the paper on Jennifer Aniston neurons. That if you
Yeah,
and that was done in humans, that if you record activity of the same like hippocampal cells, and you show them like faces of people, and they found this neuron that was like firing every time there was Jennifer Aniston in the picture, or even when like there was just a word Jennifer Aniston written, like there was no face. It was like kind of the neurons seem to prefer this particular concept, like the idea of Jennifer Aniston. Yeah, and
sorry, go ahead. I just thought of a funny example of that, but go for it.
Yeah, sure. And basically, if you think about it, this kind of space is even more abstract than sound frequencies. It's a space of like all people, basically, all people or something like that. And if you think about it, you're navigating this space of people, and if you're looking at the Jennifer Aniston picture, you are located, in this space, in the at the Jennifer Aniston point. So there is a corresponding place cell that would, fire action potentials to indicate that you are in in this location.
Yeah, that's so interesting. I think I could be mistaken, but I think it's like Quiroga and Christoph Koch maybe were involved in that research. But yeah, the one I remember was Halle Berry. They were asking them about the actress Halle Berry, and showed a picture of her, and this neuron fires. Show the name, the neuron fires, and then even show her. You can't see her face, and she's dressed up as Catwoman in the Catwoman movie, and it still fires. So it's this very like abstract conceptual thing. But yeah, that is so interesting. Very cool. All right, so I think we've now kind of we've talked about the hippocampus. We've talked about these place cells. And I guess we didn't talk much about how sleep is involved in memory consolidation. So maybe you could talk a little about that.
Yeah, sure. Because it was kind of a very long detour. But what I was started, I basically started talking about the idea of a replay and how each memory can be thought of as this sequence of place cells that are activating one after another. And when we talked about the mouse in the maze, that path that it has taken, it is essentially a sequence of the place cells that are firing that, in the beginning, in the middle, and then at the end of the path. Yeah, so you can think of it as a sequence, and then to basically to consolidate the memory and to kind of send this to more long-term storage from the hippocampus to like cortex. And this is a very elegant idea. It's called hippocampal replay, and it refers to the fact that especially during sleep, after after the waking, after like navigating, but it's also it is observed as well during rest, basically, like when a mouse is just sitting and enjoying the tasty reward. That sequence of place cells representing the path, it is replayed, in reverse order and on a much shorter time scale. So it's as if, these old like cassette players that when you rewind the tape and it's really fast and it's in reverse order. Yeah, and it seems like the brain is doing that. So it's replaying the exact sequence of place cells representing this path in reverse order, kind of to trace back the steps and to kind of send this memory like to long-term storage. And this replay is usually nested in an event called sharp wave ripple. And this is, it was given such a name due to its shape, how it looks when you record electrical activity. But basically, the activity of the network, right, because you record kind of local field potential, which you basically listen to a lot of different neurons at the same time, their collective behavior in one way or another. The activity would suddenly go up, it would increase. So it just means that a lot of neurons seem to suddenly become active and give discharges. And at the top of this basically sharp wave, yeah, there would be a ripple, so kind of very high frequency oscillation. And basically inside this oscillation, we believe is that there is this replay going on, essentially. And during these events, yeah, the brain sends information back to the cortex and consolidates the memory.
Right, and then there's experiments that you've mentioned in your video where they disrupt that activity and are then able to show that it is actually part of that memory consolidation process because it makes the mice worse than they would be at completing the maze if they didn't have that activity disrupted. Is that right?
Yeah. So basically, yeah, just disrupting the sharp wave ripple, it just leads to a worse performance the following day. So their spatial memory is disrupted, and this is at least partially what we believe is the function of these ripples, and it's one of the components of memory consolidation. So yeah, all right.
Right. So I guess the take-home message for people is, if you're trying to learn something, you have to sleep. Basically, or if you're trying to remember it, well, you have to sleep.
Exactly.
All right. So on that, I want to turn our attention to attention. So you have a video about attention, and I thought we could just talk generally about what attention is in the brain, how it works, and why it's important for learning.
Yeah, sure. And just to give a short disclaimer, the discussion about attention, since I'm not working with attention, it wouldn't be so based on neuroscience of it, the particular nitty-gritty stuff, rather than the high-level effects of attention, and especially its role in learning, as you said. So yeah, you can think of attention basically as this kind of lighthouse, and the beam that kind of this lighthouse generates like inside the brain, and it highlights the important portions of the information. Because if you think about it, at every given instance of time, you receive a ton of incoming signals, right? From your eyes, like vision, like auditory stimuli, right? Touch, smell, all those things. And you don't kind of pay attention, you don't equally process them at every given time, because how you buy new shoes, right? And like the first a couple of minutes, it feels like off and kind of uncomfortable, but then as you kind of walk around, and especially if something distracts you, like you get a text, or get a phone call on something, for some reason you don't even notice it anymore. And it's not that something is physically different. I mean, it hasn't been enough time for the shoes to physically adjust their shape. It's just that this signal, the sensory information, like from the pressure, the shoes and stuff, it's still being sent to the brain because it's a continuous process. But there is this kind of a relay station, a structure called thalamus, and it essentially receives all of these information, all of pieces of information, right? Of the channels, things like visual stimuli, basically all senses, except olfaction. For some reason, smell, it just bypasses the thalamus. But and thalamus then kind of decides what to let up into the cortex for further processing, and what is not worth, and this kind of selection process is at least partially what we call attention, at this stage. And there is also cognitive attention, which is like probably in prefrontal cortex. It's like, it just refers to when you're watching a lecture and you think of something else, like what to order for dinner, right? Information from the lecture, even though you receive it, it makes it through. It makes the way through the thalamus. It's just in the cortex. It's not highlighted. And I'm sure there are more like attention systems like these in different areas and sub areas that we haven't identified, like their mechanisms and their location and stuff like this. But it's more of a concept, let's say, how the brain works. It seems to be somewhat fundamental. And both these types of attention, both the at the level of thalamus, and especially the attention that's going on, this attention beam in the prefrontal cortex, it is really important. It's vital actually for learning, because if the information is not highlighted, well, it has almost no effect on the circuits. Like there is, like, you don't do any active process. Like, again, I don't want to go into I don't want to say that there is no synaptic plasticity, because I'm no expert on this, and probably there is some plasticity, but the high level effect is that you don't learn anywhere as effectively as you would learn something that you're devoting your full attention to.
Right. So anybody who thinks that they can just listen to an audiobook while they sleep and they'll somehow absorb all the information is probably mistaken, because it seems obvious to say it, but you have to pay attention to actually learn something effectively.
And one thing that's crucial about attention, because and I think this is often overlooked, that this highlighting beam, let's say, right, that you select different portions of the information, you can't move it instantaneously, right? So there is some inertia in doing so. So when you kind of switch your attention from one task to doing something else, right, kind of your cognitive efficiency is decreased, so to speak. You can think of it as a car engine, for example, that when you are running at full speed, and then you completely stop, you shut down the engine, and then for you to kind of to start moving again, you have to, start the engine, you have to accelerate, stuff like this. So it's really similar, when it comes to attention, because you can't instantaneously switch between contexts and between tasks. And this is fundamentally why multitasking in conventional understanding is not it's not effective, and it's quite harmful. And this is why it's important to eliminate all sorts of distractions when you study, or when you're like doing something cognitively with high cognitive load, for example, like coding or reading, because it might not seem like a big deal. I mean, you're watching a lecture and your friend like texts you, so you check out the message real quick, or just give it a glance, and then you look back at the lecture. But down inside the brain, like, you can't do this process instantaneously. So how you perceive the information would not be as effective. How you process it. And I think this is very crucial. And usually people think that they can multitask and switch contexts on and off, but it's not the way it works.
Yeah, it degrades your performance on both tasks that you're trying to do. Exactly. I talked to the neuroscientist David Better, who studies cognitive control and working memory and things like that. And he talks about one way to see this is, if you, like, your car analogy was really great. Another way to think about it is, if I were to just say the whole alphabet, A, B, C, D, all the way to Z, and then I were to say the numbers 1 through 26, so, it's the same number of characters. I could do those really fast. 1, 2, 3, 4, 5, 6, 7, 8, go all the way to 26. A, B, C, D, E, F, G, H, go all the way to Z. But if I were to switch between the two, going A1, B2, C3, D4, E5, like, even just then you can see me having to think, okay, what's the fifth letter? So it's just this way of showing that when you're switching between the tasks, your overall task time is going to be longer for trying to switch than if you just added the 2 times for saying just the letters or just the numbers. So a little bit different, but sort of the same kind of inefficiency of multitasking.
Yeah, I really like that analogy as well. I think it's really clever.
Yeah, definitely. All right. Well, I think I want to zoom out a little bit. And so I've been asking all my guests, everybody who studies neuroscience to some extent, to give me a short answer to this kind of ridiculously general big question, but it's, what is your short answer to how does the brain work?
Oh. Well, I'm sure you're not going to, let me off the hook with saying we have no clue, which is why I'm going to speculate.
But that's the honest answer. We have no clue, right?
Yeah. I think the brain, like, the main job of the brain is to kind of use the incoming information to predict what's going to happen. So to construct this model that's constantly updating the model of the external world. I think this is called predictive coding. Really nice concept. Essentially the only reason why such complex brains emerged through evolution is that we needed some fancy way to to carry out behavior that would lead to, in the end, to more effective reproduction, because that's the point of evolution. And on that path, constructing the model and predicting, using the stimulus, basically the history of past stimuli, to predict what's going to happen and construct an action. And I think this is really important, because the brain is not a passive receiver. Like, its main job is to generate effective actions, because if you think about it, the animal that can perceive but cannot move, in this case, the perception would be completely useless. Mm-hmm. However, if the animal cannot perceive anything but can randomly move, it would be more effective in terms of survival and reproduction, because there are at least some action. And the goal of the brain is to use this model, which we don't know how it happens, but to come up with actions and execute them that would, lead the organism to more effective reproduction.
That's a good answer. That's a solid one. All right. Well, so some of the other questions I've been wanting to ask people who are in this field, and I think which are of interest to many people who are interested in the brain or the mind or even philosophy. And feel free to say no comment if you don't want to talk about them, don't want to answer them. But what is your view of free will?
Second.
Gotta take a drink of water.
Yeah, that was my act of free will to take that sip. I believe that, free will exists. And, one argument I hear most often about free will is how they kind of deconstruct that everything is determined through, if you look at atomic interactions and everything perfectly, how you can calculate, what's going to happen on a macro scale. I don't buy that. To me, this I can try to remember it's sort of like this famous paradox about the turtle and what's the very fast guy called in English? I don't remember.
What was the hare, the rabbit?
I was aiming for the human, the muscular one, but the rabbit works as well. It doesn't matter. Basically the paradox is that, if you have like a turtle and then you have a rabbit start, like let's say 100 meters behind it, and they have to run a race, like let's say 500 meters or something. And, you ask who is going to be the winner in this case. And the intuitive thing, and what's actually, what's obviously is the case in reality, is that rabbit, since it's like 100 times faster, it will outrun the turtle and, reach the finish. But there is this famous paradox that since the turtle starts 100 meters ahead, by the time that the rabbit reaches the point where the turtle has eventually started, so by the time rabbit runs these 100 meters, the turtle would have moved a tiny, teeny bit, let's say like a few centimeters. And then for the rabbit to cover this, this new distance of a couple of centimeters, it would take some time. So it would be like very short, but still amount of time. And so by the time rabbit reaches that point, when the turtle was, it would still have moved like a ridiculously tiny amount of distance, but it would still have, it would still, be ahead. And, you make this argument that if you repeat this, until the infinity, so there would still always be some distance behind the rabbit and the turtle, and that rabbit would always be behind. And so basically conclude that the rabbit will never reach the turtle, which is obviously not the case in reality. And one explanation is that you cannot infinitely divide the continuum of time in this case. And I don't know why, but we actually had a discussion during a summer school when I was, I think it was in 10th grade. We had this discussion about free will, and I heard the argument about how you can like divide the time and move like kind of closer and closer to say that everything is essentially like predicted. I found it very reminiscent of this paradox, because you kind of infinitely divide and you look at infinitely small distances, small scales, both in case of space and time, which could to your brain result in this kind of paradox, right? And do you see the point that I'm making? That
I think so. Yeah. That, I mean, it's, we can try to break down behavior and the physiology of the brain down to just the movement of atomic particles and molecules and the chemistry of what's going on. And then therefore we could predict that behavior of the system if we knew the position and velocity of every particle. But what you're saying is that that may be a fallacy, because as we get closer and closer, we just may not be recognizing some other kinds of principles that are operating that do allow for some kind of free choice or free will to arise. Is that kind of
yeah, that's a high level conceptual. It's more of an analogy between this paradox. And another argument is that first of all, it's impossible to measure obviously all the velocities and all of the positions, because there is an uncertainty principle, right? You can't know everything about particles, and there are different quantum effects and fluctuations and stuff like that. And even if you can't measure anything perfectly, and we have a very good rationale to believe that a brain or biological processes can be an example of chaotic systems. So systems which are very sensitive to initial conditions, right? A slight nudge in in initial conditions, right, could result in a totally different behavior. And since you cannot physically measure everything perfectly, you cannot know everything, you cannot predict. But this is more of a case when if one would actually try to to measure everything and to predict, that wouldn't be possible. But so there's those two kind of explanations, one more conceptual understanding, and the other one has to do with physical limitations and processes of dynamical systems.
Right. All right. Well, that's a good answer. That we could probably go on for another hour about that, I think. But anyway, Artem, I really appreciate you coming on and talking with me today and answering my questions. I know some of them maybe got a little bit out there, but I really appreciate you just answering all those and talking with me today. So thank you.
Thank you. My pleasure. I totally enjoyed the conversation as well.
Great.
Yeah. If you want to do it one other time down in the future, don't hesitate. Yeah,
I'd love to
drop an email. Yeah, I totally love to repeat that as well.
Great. We'll keep it up then. All right. Well, maybe you can. I'll definitely link to your channel and everything in the description. But maybe you can tell people where to find you, what your channel is called, and all that.
Yeah, sure. My channel is called by my real name, so it's Artem Kirsanov. Hopefully, this isn't so distorted because it's Russian language. But in English, if you basically it's spelled as it sounds. I'm quite a lucky person in this regard, because I know many people who are, whose names and surnames, you listen to them in Russian and then you try to guess how it's spelled in English, and like it's a possible job. And sometimes they actually take like 3 times as much space due to sounds that are unique to the language. So you have to like combinations of letters. But yeah, and like Andrew said, down in the description, probably, there is a link, or you can search for the famous neural manifolds video, and it will surely pop up right at the top of the search.
Great. All right. Yeah, and your name will be in the title of the video. So anyone wondering, look no further.
Yeah, that's fair.
All right. Well, thanks again. All right. Well, that is it. Thank you so much for listening or watching this episode of the Thinking Tools podcast from Sense of Mind. Be sure to like and subscribe to the YouTube channel and to the podcast. Also, consider giving this show a 5-star rating on whatever podcast platform you use. As always, this channel is brought to you by the Diamond Mind Foundation. This episode was produced by me, Andrew Cooper Sansone. Thank you so much for watching. I'll catch you next time.